Hello, my name is Emilia Hurtado, and as it was stated, I'll be talking to you about MiClone today, a probabilistic method for inferring self-phylogenies from mitochondrial variants. All right, so since we'll mostly be talking in a cancer context in this talk, we'll start with a bit of cancer background. Typically, when a cancer starts, what you have is a founder cell, like this right here, that gains an oncogenic change, which really just means it's gained a change that allows it to start proliferating uncontrollably. And that will proceed as the disease progresses, such that those daughter cells are also going to be proliferating uncontrollably. And over time, they too will be picking up their own changes. What this ends up causing is the rise of these distinct but related clonal populations of cells. And the reason that this all matters is because usually by diagnosis, a patient's tumor is not, in fact, one homogeneous mass of cells. It's, in fact, a heterogeneous mixture of these clonal populations. And those clonal populations might just respond differentially to treatments. For example, in this graphic here, we can see we've got our founder cell. It's proliferating. It has given rise to these clonal populations here, and here we are at diagnosis. At this point, they apply the therapeutic, which is rather effective, save for the fact that one clone was not sensitive to that therapeutic. The result of that is after some amount of time, whether it's months or years, that one clone might also expand and cause its own clonal expansions to happen. And then that first therapeutic that worked so well the first time will not be effective since that clone was already not sensitive to it. So this kind of highlights why it might be important moving forward that we understand the clonal composition and dynamics of a patient's tumors. So much work has actually been done to characterize this problem to, say, from bulk data, deconvolve, that is, figure out what the actual clonal mutational profiles are going to be, and then build the phylogenetic tree, that is, see how those clones are actually related in bulk data. So we have various methods that perform very well. They each improve on the state of the art. And now we have the advent of single cell sequencing, which allows us to study clonal dynamics at single cell resolution. Unfortunately, both of these methods still fall prey to one limitation, the fact that they both rely on genomic instability. What that leads to is that genomically stable cancers, so that would be cancers that can't be resolved by copy number, maybe they have very few somatic mutations that are driving them, are more difficult to study. There are currently options. You could go about it by doing a targeting sequencing of a bespoke mutational panel. Downside of that is it's tedious, time-consuming, it might be costly. You also have to know the mutations ahead of time. Alternatively, you could do targeted sequencing of a pan-cancer mutational panel. The downside here is typically the driver mutations included in those panels tend to be clonal, which doesn't give you much information to actually resolve subclonal dynamics and relationships. Now, recently, there's actually been efforts to leverage the mitochondrial genome and the variants within to actually build cell lineage. And it makes sense to do so given specific aspects of mitochondria. For example, any one human cell is host to hundreds to thousands of individual mitochondria. And the mitochondrial genome itself happens to have a much higher mutation rate than the nuclear genome. Furthermore, it exhibits heteroplasmy. So that's the concept that, yes, while the cell is host to many different mitochondrial species, there's nothing to say that those mitochondrial species are all identical. In fact, what we may end up having is a cell that has multiple mitochondrial clones populations in that one cell. And lastly, the mitochondrial genome is small. So getting high coverage sequencing doesn't necessarily require amplification. And there's also been advancements in mitochondrial methods. For example, there are specific sequencing methods that are coming out to capture more of the mitochondrial genome as they're being sequenced. For example, the mitochondrial enriched version of single cell ATAC, which additionally can allow for functional or state analysis of cells, which lets us hook in the genotype with phenotype. We also have a couple methods that work at identifying important or informational mitochondrial variants. For example, we have MGAK, we have MQuad. And at the time of the writing of this presentation, there is at least one that does attempt to take those variants that were identified and then build the clonal and then cell lineage tree from mitochondrial variants. Fortunately, there is still a limitation that we've noticed here, that they all underutilize the signal present in cellular heteroplasmy. So then the question is, like, how would we use heteroplasmy as signal? For example, we have this one cell here. It's got 3 distinct clonal populations of mitochondria. When the cell divides, they will double in number. And then when they split out to the daughter cells, although the actual number that splits out will be equal, there's no guarantees about what the actual mix to each daughter cell will be. So what that results in is something like this, where you have the 2 daughter cells with these somewhat different mixtures of the clonal proportions or clonal prevalences. So then the question might be, how are we going to take this and use it to actually build a cell lineage? We actually approach the problem from 2 stages. So the first stage of that would be the mitochondrial clonal deconvolution problem. So that's to say, if I'm given from all the single cells variants that are called just for mitochondria, how do I take those and their read counts and then produce a clonal phylogeny and a per cell measurement of the clonal prevalence estimates in each cell? So that is how much of each clone is present in each cell. And the second problem is actually making a lineage from that. So if we assume that we've solved the first problem, we're given now the mitochondrial clonal phylogeny, and we have, of course, the per cell clonal prevalence. How do we take both of those things and then actually produce the cell lineage tree? So at this point, I'd like to introduce yet again MiClone, a probabilistic method for inferring cell phylogenies from mitochondrial variants. And we'll just kind of walk through how MiClone will map against those 2 problems we just outlined. So we start here. Again, we've got the mitochondrial variants and their read counts from each single cell BAM. We've done the variant calling there. The next step then is to feed that through to PyClone VI for pre-clustering and early cellular prevalence estimates. From there, it is put through MiClone, which will in fact do the work of building out the clonal phylogeny and establishing what the clonal prevalence estimates are. And finally, MiClone's last step is to take a tree distance metric that can use both the information present in the clonal phylogeny and take, of course, those per cell clonal prevalence vectors and use them to output the cell lineage. So before we get into what the actual results are with the early benchmarking there, a bit of background information about the actual data that we're working on. So we benchmarked MiClone's current implementation against 22 real patient data sets across a variety of cancer types, which include high-grade serous ovarian cancer, triple-negative breast cancer, follicular lymphoma and its transformed partner, diffuse large B-cell lymphoma, and follicular lymphoma again. So each patient data set was sequenced using DLP+. That is a very scalable single-cell whole genome sequencing protocol, which provides us with a bias-free single-cell whole genome data set. And we would also call copy number, total copy number for each cell such that we can build a total copy number tree. This total copy number tree is what we're going to actually use as our orthogonal feature to compare the methods inferred trees against to get an idea of how accurate it's being. And as for the actual metric we're using to define phylogenetic accuracy, we're going to use the generalized Robinson-Folds distance. So what that actually is it's a metric that's going to tell you how far your prediction actually is from truth. So if you're very close, your number's lower, and that's good because you're close to truth. If you're very far away, less so. The first set of results that I'm going to present here is actually just an internal comparison. So MiClone is very much still in development. So we had to kind of decide what tree construction kind of protocol we wanted to go with for that cell lineage tree. And here you can see the comparison of the different distance metrics that we used and the different types of tree construction kind of ideas that we had, right? So we tried neighbor joining, UPGMA, weighted UPGMA, and ultimately found that the neighbor joining plus tree Wasserstein distance was the most informative. So in the next set of results, when MiClone is referred to, it will be referring to the neighbor joining tree Wasserstein construction of the tree. The next thing we wanted to do is make sure that we would perform well against naive baselines. When I say these baselines, it's basically the concept that we take just a very simple way of building the tree and see how we perform against that. We included 2 versions of these baseline trees. The first is the variant allele frequency tree, or the VAF tree, where we just take the VAF for the mutations across the cells and we use that to build the tree. The second one is cellular prevalence baseline tree, which is very similar to VAF, except instead of using the VAF, we're going to use the cell's cellular prevalence for each of those mutations. And here you see that MiClone does tend to outperform the baseline trees. So again, lower is better. Okay, so again, at the time of writing, there was just Merlin to compare ourselves to. And just a quick note, the variants that were provided to Merlin were run through MQuad first. That was due to the fact that the author's information on the actual repository stated to do so, which is exactly what we did. We took our data, we ran it through MQuad, we provided those variants to Merlin. And here I've broken it apart by the different cancer types to see how we each do across them. And as you can see, MiClone outperforms MQuad plus Merlin across the cancer types. And finally, I'd like to present a result that kind of shows how MiClone can also approximate like robust results from previously published works. On the left here is a Sitka tree, which is a method that will run on your nuclear DNA variants, and it will kind of use breakpoints to establish what the phylogeny is. And here's MiClone, and using only the mitochondrial variants and mitochondrial signal available in the same set of cells, you can see it is approximating the same result that Sitka is with just mitochondrial signal. What you're actually looking at here, by the way, are copy number heat maps. So each chromosome, and then when there's an actual event, you can see that this blue one would be a loss, the yellow is a gain. So like I said, MiClone is very much still in development, but these early results give us confidence that it could be the approach is worth actually pursuing. And to that end, I'd like to thank my supervisor, Andrew Roth, Dr. Alexandre Bouchard-Cote, everybody in the Roth Lab, and of course, all the funding agencies that support our efforts.
Hi, lovely. How can you be sure that you see all the mitochondria in one single cell? Because we have this kind of sparseness even within a cell. So how is that?
Very fair question. That's actually, maybe I actually forgot to mention it. That's one of the reasons why we're using data sets with so many cells, because we're performing this across thousands of cells. We're trying to deal with the fact that, in fact, yeah, you might not have it all, but if you have enough of them, you probably will. The numbers. Also, DLP is really good at picking up mitochondria.
Yeah. Thanks. So it's clear that even for mitochondria, the mutations are not neutral, right? So I mean, some mutation may have an advantage in terms of proliferation of the mitochondria itself, right? Division of the mitochondria itself. Leading to overexpansion of some specific mitochondria with some specific mutations. Is it some bias do you consider, or do you think it may be a problem for you?
I feel like since we're actually charting those exact proportions to figure out how the cells were dividing over time, like generationally, I at this point don't see too much how it might affect us, but it's a very good thing to take into consideration.
Thank you for the nice talk. I had two questions. So the first is that it seems like the problem of reconstructing the clone tree is kind of equivalent to doing bulk DNA sequencing, tumor phylogeny reconstruction. Like, is that a reasonable baseline to use?
100% true, yeah. Yeah, like we're viewing each single cell as a bulk sample of mitochondria.
Exactly. Of course, there's some assumptions baked in there. And then the second question is on these RF distance plots. How is that RF distance normalized? Because they seem quite high. Like, I don't know, depending how you normalize, it can be between 0 and 1, and these were like
it would be between 0 and 1.
Yeah. So then the RF distances seem quite high, meaning that you're, like, almost all the edges are wrong. So that's
yeah, it's still in development. Yeah, we have a few where we are quite a bit lower. Also, there are a number of cancers there that are maybe a bit harder for us to actually function well on. And it's a good thing to keep an eye on. And as we develop, hopefully we'll improve.
Cool. Thank you.
Hi, thank you for the wonderful talk. I just wonder, what do you think about the opportunity for your method to work with other methods for single-cell whole genome sequencing, like PTA or something like that? Not MDA for sure, but just something more linear.
The way I look at it, if you can call them and they're single cell, it should work.
Okay, great. Thanks.
Do we have more questions here in the back? There's a seat there. There's a seat there. There's a seat there. Actually,
Oh, there's online question.
Yep.
Yes.
oh, good. Your last slide shows that the lineage deviation point according to nuclear and mitochondrial deviation. Why do you think they're connected?
Sorry, say again?
It says your last slide shows that the lineage deviation point according to nuclear and mitochondrial deviation. Why do you think that they're connected?
As in the construction there, like, why do I think them being similar? Well, the idea is we're trying to see if just by using mitochondrial signal, we can also capture the same kind of events that are captured just by the copy number changes. Trying to use it as an orthogonal feature to see if it's at all functioning.
I have a question which is similar to the one that Henry asked, which is, it seems to be like basically you're banking on the idea that you have a population of mitochondria and that your within population variation is going to be smaller than your cross population variation between this group of mitochondria and this cell and this group of, is that basically what the right way to think about it in terms of, do I have information? If I compare these two groups here with this group here, am I going to actually get a phylogenetic signal?
So the question is, you're asking,
Well, so you have all these mitochondria, they're all independently experiencing mutations.
yes,
So, and then this group over here is also independently experiencing mutations.
Mhm.
So they somehow have to be more different across cells than within cells.
ultimately, yeah, we're just trying to, again, use the proportions of the mitochondrial species that are there. So if we can define what the clones are, then we chart the proportions as they move. It could be that it is higher in some, or that some have no representation of some clones, some have a lot of the others. The idea is to use those actual distinct different proportional profiles
Mhm.
to build out the signal.
Thank you.
There are no more questions. Then let's thank our speaker again.